Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Exploring techniques and tools for whole genome. Sequencing of bacterial genomes.
Welcome to our demonstration of whole genome sequencing tools, where we'll showcase the powerful analysis techniques used for species identification, MLST typing, and detecting resistance genes in bacterial genomes. Through this demonstration, we'll highlight how these tools empower researchers to accurately characterize bacterial species, track their evolutionary paths, and uncover crucial insights into antibiotic resistance.
Objective of this tutorial. 1. Identification, which has determine species using Kmer Finder, identify sequence type, ST, using MLST. 2. Plasmid analysis, detect and type plasmids using Plasmid Finder and PMLST. 3. Resistance gene, identification, use ResFinder to locate resistance genes. 4. Phylogenetic relationship, construct phylogenetic tree with CSI Phylogeny. 5. Decision-making, compile results in Excel. Determine 3 outbreak strains based on species, ST, resistance genes, plasmids, and phylogeny. Let's start with knowledge about the tools and techniques, starting with Kmer Finder. When encountering bacterial organisms in our research, a fundamental question arises. What species is it? Traditionally, the 16S rRNA gene has been pivotal for sequence-based taxonomy, but it does have limitations. To overcome these limitations, alternative methods like utilizing the GIRB gene, ribosomal MLST, or specific protein domain profiles have been proposed. However, why limit ourselves to small gene fragments when we have access to the wealth of genetic information provided by whole genome sequencing data? The Kmer approach, harnessing whole genome data. Whole genome sequencing offers comprehensive genetic insights. Instead of fixating on isolated genes, we can harness the entirety of genetic data encoded in the entire genome. This is where Kmer Finder comes into play, a tool that utilizes Kmer's short DNA sequences to infer species from whole genome sequencing data. Understanding Kmer's. Let's unpack Kmer's. These are short DNA sequences with a defined length denoted by K. For instance, a 10-mer represents a sequence of 10 base pairs. By segmenting the genome into these Kmers, we can compare them across different organisms. Database and comparative analysis. Kmer Finder relies on a database containing Kmers from known species sourced from NCBI. When presented with an unknown genome, the tool dissects it into Kmers and cross-references them with those in the database to pinpoint the best matching species. Efficiency optimization. To expedite processing, redundant data in the database are minimized. For example, only specific Kmers with particular prefixes may be retained. This optimization ensures swift and efficient species prediction. Performance assessment. Extensive testing has been conducted to assess Kmer Finder's performance. Comparative analyses showcase its superior accuracy in predicting both genus and species levels compared to existing tools. Result interpretation. When analyzing results, focus on the best matching species and consider parameters such as Kmer score, template length, template coverage, query coverage, and depth. These metrics provide valuable insights into the reliability of the prediction. Example, parameters for interpreting Kmer Finder. Results, interpretation. In this example, Escherichia coli is identified as the best matching species with a high Kmer score, 174,000, and significant template coverage, 99.83%. The query coverage is also substantial, 96.98%, indicating a strong match between the query and reference genomes. Additionally, the depth is close to 1, 0.99, further supporting the reliability of the prediction. This collectively suggests a high confidence in identifying the species as E. coli. Conclusion. In summary, Kmer Finder revolutionizes species identification by harnessing the power of whole genome sequencing data and Kmer analysis. Its accuracy, efficiency, and accessibility make it an invaluable asset for researchers across diverse fields. Next tool is MLST tool. After finding Kmers, short sequences of DNA bases of length K, multilocus sequence typing, MLST, is used to characterize and classify bacterial strains based on the sequences of specific genes. Here's why MLST is a valuable tool after identifying Kmers. Higher resolution. While Kmers provide a glimpse into the genomic content and diversity, MLST offers a higher resolution by focusing on specific genes. MLST targets conserved housekeeping genes across bacterial genomes, providing a standardized method for comparing strains. Standardization. MLST targets a set of housekeeping genes that are universally present across bacterial species. This standardized approach ensures consistency and comparability of results across different studies and laboratories. Discriminatory power. MLST can provide higher discriminatory power compared to Kmer analysis alone. By analyzing the sequences of multiple loci, MLST can differentiate closely related strains that might have identical or very similar Kmer profiles. Epidemiological studies. MLST is widely used in epidemiological studies to track the spread of bacterial strains, understand outbreaks, and study the population structure of bacterial pathogens. The sequence data generated through MLST can be easily shared and compared between different research groups and public health agencies. Evolutionary analysis. Data can be used to infer evolutionary relationships among bacterial strains. Phylogenetic analysis of MLST sequences allows researchers to reconstruct the evolutionary history of bacterial populations, understand the genetic diversity within species, and study the mechanisms of adaptation and evolution. Clinical applications. MLST data can have clinical implications, such as identifying antibiotic resistance patterns or predicting virulence potential. Understanding the genetic makeup of bacterial strains through MLST can help guide treatment strategies and inform public health interventions.
Overall, while Kmer analysis provides valuable insights into genomic composition and diversity, MLST adds another layer of information by focusing on specific genes relevant for strain typing, epidemiology, and evolutionary analysis. Combined, these approaches contribute to a comprehensive understanding of bacterial populations and their dynamics.
After performing multilocus sequence typing, MLST, another tool commonly used in bacterial genomics and epidemiology is ResFinder. ResFinder is specifically designed to identify acquired antibiotic resistance genes within bacterial genomes or plasmids. Here's how ResFinder fits into the workflow. ResFinder for antibiotic resistance. Following MLST, ResFinder is utilized to identify acquired antibiotic resistance genes within the genomes or plasmids of the characterized bacterial strains. These resistance genes confer resistance to various antibiotics and are crucial for understanding antibiotic resistance profiles and patterns. Clinical and epidemiological relevance. ResFinder results have important clinical and epidemiological implications. They provide insights into the potential antibiotic resistance profile of bacterial strains, which is critical for guiding antibiotic treatment strategies in clinical settings and for monitoring the spread of antibiotic resistance in the community and healthcare settings. Public health interventions. Information from ResFinder can inform public health interventions aimed at controlling the spread of antibiotic-resistant bacteria. By understanding the prevalence and distribution of antibiotic resistance genes in bacterial populations, public health authorities can develop strategies to mitigate the impact of antibiotic resistance on human health. Research purposes. ResFinder data is also valuable for research purposes, such as studying the mechanisms of antibiotic resistance, tracking the evolution of resistance genes, and investigating the genetic basis of antibiotic resistance in bacterial pathogens. In summary, ResFinder complements MLST by providing information about antibiotic resistance profiles in bacterial strains. Together, MLST and ResFinder contribute to a comprehensive understanding of bacterial populations, their antibiotic resistance profiles, and their epidemiological dynamics, which is crucial for both clinical management and public health interventions. After utilizing MLST and ResFinder, Plasmid Finder is another tool frequently used in bacterial genomics and epidemiology, especially in the context of studying mobile genetic elements like plasmids. Here's how Plasmid Finder fits into the workflow. MLST and ResFinder results. Before using Plasmid Finder, researchers typically have results from MLST, which provides information about the strain type, and ResFinder, which identifies acquired antibiotic resistance genes within the genomes or plasmids of the characterized bacterial strains. Identification of plasmids. Plasmid Finder is then employed to identify and characterize plasmids within bacterial genomes. Plasmids are extrachromosomal genetic elements that can carry a variety of genes, including antibiotic resistance genes. Identifying plasmids is important because they often play a significant role in the horizontal transfer of antibiotic resistance genes between bacterial strains. Understanding plasmid dynamics. By identifying plasmids, researchers can gain insights into the dynamics of horizontal gene transfer and the dissemination of antibiotic resistance genes within bacterial populations. Plasmid Finder helps in understanding the prevalence, diversity, and distribution of plasmids among bacterial strains. Association with antibiotic resistance. Plasmid Finder results can be correlated with the antibiotic resistance profile obtained from ResFinder to investigate the association between plasmids and antibiotic resistance. This information is crucial for understanding the mechanisms underlying the spread of antibiotic resistance and for developing strategies to combat it. Research and epidemiological studies. Plasmid Finder data is valuable for various research purposes, including studying the evolution of plasmids, tracking the spread of plasmid-mediated antibiotic resistance, and investigating the role of plasmids in the adaptation and survival of bacterial pathogens in different environments. In summary, Plasmid Finder complements MLST and ResFinder by providing information about the presence and diversity of plasmids within bacterial genomes. Together, these tools contribute to a comprehensive understanding of bacterial populations, their antibiotic resistance profiles, and the dynamics of horizontal gene transfer, which is essential for both research and public health efforts to combat antibiotic resistance. CSI Phylogeny is a tool used to construct phylogenetic trees based on whole genome sequence data of bacterial isolates. Here's how CSI Phylogeny works and how it helps to determine how strains are related. Whole genome sequence data. CSI Phylogeny takes whole genome sequence data of bacterial isolates as input. These sequences contain information about the entire genetic makeup of each isolate, including core genomic regions as well as accessory genetic elements like plasmids. Identification of core genomic regions. CSI Phylogeny identifies core genomic regions shared among the isolates. These regions typically represent conserved portions of the genome that are present in all isolates. By focusing on core genomic regions, CSI Phylogeny can ensure that the phylogenetic analysis is based on stable and reliable genetic information. Phylogenetic tree construction. Using the identified core genomic regions, CSI Phylogeny constructs a phylogenetic tree that illustrates the evolutionary relationships among the bacterial isolates. The phylogenetic tree depicts how closely related or distant the isolates are from each other based on their genetic similarity. Visualization of relationships. The resulting phylogenetic tree generated by CSI Phylogeny provides a visual representation of how strains are related. Isolates that cluster together on the tree share more genetic similarity and are considered to be more closely related, while isolates that branch apart are more genetically distinct. Interpretation of results. Researchers can interpret the phylogenetic tree generated by CSI Phylogeny to understand various aspects of strain relatedness, such as the genetic diversity within a population, the evolutionary history of the isolates, and potential transmission pathways in epidemiological investigations. Now let's start with practical assignment. I will give you a file which will contain 5 unknown samples and an Excel file to summarize your results. In the end, we will identify 3 strains which are outbreak strains. Let's start.
Excel file will have an empty table where you will have to fill your data. Here you can see all the antibiotic classes and plasmid column with MLST. Go to a suitable browser and type in Kmer Finder, and then click the second link. Here you can see the interface of your tool. It has instruction on how to use it and how to read the output. You can also download the software version as well. So you need to upload single FASTA file or 1 or 2 FASTA Q files. You can read the sequence as per your sequence database. Now choose the file which I have given you. Make sure to unzip it before using. I have selected the first strain and uploaded it on the tool. After a few seconds you will get this screen. Don't close the tab and keep this screen open. It will show you result on this interface, or you can write your email and it will email it to you. Here you can see I got my result. As I have already explained you how to read the results. We have got 2 strain match, Escherichia coli and Salmonella enteritica. So we have to see which species our unknown sequence matches completely or partially. You can see the query coverage, template coverage and depth as well, and the difference in between both of the species. You can go into in-depth by clicking Extended Output. And with that you can see all the information and even the accession number. You can use the accession number in various databases, and as well as you can see there is text ID and text ID species as well. And lastly, species column. Now we'll see which one matches the sequence we uploaded. It was Escherichia coli, and we will put the name into the Excel sheet. So this was it for the Kmer Finder tool. You will repeat all these steps same to the other strains which I have given you and paste it into the Excel sheet for your result. You can see I have collected all my unknown sequence species, and now I will head to ST type. You can go into the same interface and click on the services and scroll down to find MLST type finder. Here is the interface of the tool. You can get to understand its instruction and output. Make sure you read everything on the tool. Now by clicking on the select button, you have to select Escherichia coli 1. You can leave all the settings at default unless you want to go in depth and you have an understanding of other options as well. Read carefully all the instruction on the tool. It will tell you which file to upload and what options you have to select. Here I have selected Escherichia coli 1 and I will select my strain and upload it on the tool. Same goes for this. You can type in your email and it will notify you on email. Or you can keep the screen open. And here are the results. I have got my sequence type. Now I will copy this sequence type and insert it into my Excel sheet. And I will repeat this step overall to other strains as well. Here are the results which I got from that MLST tool. Now I will head to ResFinder and find out what are the resistance genes which are included in these strains. You will find the ResFinder in the Services page in the Phenotype section. And here you have an interface of the ResFinder. You can keep everything on default unless you have an understanding of what these data are talking about. Scrolling down, I will type in my email address and upload my sequences. You can upload 2 sequences at a time. And then click Submit Job. For this, you will get the results within few seconds. So here are the results. You can see all the classes of antibiotics and which one are resistant and which are not. You can even see acquired AMR gene hits. This will give you an understanding based on your research subject. It is a very useful tool. It will help you eliminate the bacteria which you have identified. If you scroll down more, you can see all the gene alignment as well. If you scroll down, you can see download options which you can add into your research paper. Heading back to the top, click on the column and it will organize for you all the resistant genes on top of the list. And then you can copy and paste which genes are present or which are not. Here, type in resistant and non-resistant. These are the collected results. Now let's head to Plasmid Finder. On the same page, you will also find Plasmid Finder by scrolling down into typing. Unfortunately, it has only 2 databases regarding gram-positive and Enterobacteriaceae. So we know our bacteria belongs to this group and we will upload our strains. Keep all the settings to default and upload it. After a few minutes, you will get your results on email or on that same screen. As I told you before how to read this data, we can see the query and template length and what are the differences between each template to the query. By clicking on the Extend Output, you can see all the differences in between the templates. Anyhow, for this assignment, we have to see, are there any plasmid present in our strains? We will type in present and do the same sequence to all the rest of the strains. And here are the results. So you can see when you have no plasmid, it will give you a data of such type. Now let's head to CSI Phylogeny. This tool will tell you the relatedness between each of the strains. I have the reference genome as well uploaded on the drive and as well as all the 5 strains to which you can compare. If you have a deep understanding of other options, you can explore and use it as well. But now I will keep everything at default and upload it. You can type in your email or keep this browser open for the results to be opened. But unfortunately, the website is loaded, so it will tell you the results a bit later. For the moment, you can check your phylogeny tree by uploading Neovic sequence in this. You will go into the files and then click on the desired file which is in NIC format and copy your data. Then you will paste it on here and then click on load. You will see the difference between all the 5 strains. Here you can adjust the values to make the graph a little visible to read. Then we have another tool which is Toll, where you can also do the same and upload all the sequence and then view the phylogeny tree. This is a bit much more better than that one that I previously showed you. Here you can see the relativeness and closeness and the distance between all of the strains from each other. You can adjust the table to read it more better. So now let's head to the practical assignment that we will solve by using the results that we have concluded from all those tools. So what is the specimen of the unknown samples? It is Escherichia coli. Our next question is, which sample have the same ST type? You can choose any one of these. You will check which one have the same ST type and then click that as an answer. Next question, which unknown samples contain plasmid? Here in the table we have written present and not present. You have to check which one was in the present ratio and which one was in the not present ratio and select your answer. Next, which sample do not contain gene conferred to the beta-lactam resistance? You have to check which one was resistant to beta-lactam. So here I will mark those answers which are not resistant to beta-lactam. Next, why do you need a reference genome for construction of SNP phylogenetic tree? So my answer is SNPs, which are known as single nucleotide polymorphism, which are always different in all the species. You can identify your species by this. Next, what unknown sample is the most distantly related strain? So our answer is 4. As you have seen in the tree, 4 was distant. Next, from the answer of question 6, why is the strain the most distantly related? So my options will be based on my knowledge. Which unknown samples are outbreak strains? So antibiotic microbial resistance is the most dangerous outbreak we will face in the coming years. So the more resistant the bacteria is to the vast or broad amounts of antibiotic, the more dangerous it can cause outbreaks. So we will select those which are most resistant to the antibiotics. Next question, from the question 8, why did you make decision that those strains are outbreak? You will analyze your result by the table and select the options which are suitable. And lastly, one of the strain has the same ST type as the 3 outbreak strains. Why is that strain not the part of the outbreak? Analyze your data and then choose your correct answer. That's it for today. This is how you identify your unknown sequence and configure your outbreak strains. Thank you so much.